Lab 3. This lab focuses on understanding the role of lambda phage. These phages infect bacteria and inject their DNA into the cell. Once the DNA is in the bacterial cell, some of the phage genes can be turned on. The phage DNA then follow either the lytic or the lysogenic pathway. Using the lambda vector allows substantial amounts of the DNA of interest to be grown up and released from bacterial cells. Consequently, knowing the titer of a suspension of phage, or the number of infectious phage particles per milliliter of suspension, aids in the proper use of the phage. Part 1 of this lab will demonstrate how to properly titer a suspension of lambda phage. Some of the materials that you will need for this lab are as follows: melted soft agar, 6 dilution test tubes, each of which individually labeled, E. coli, suspension medium, phage stock, a permanent marker, phosphate buffered saline or PBS, micropipette tips and micropipetter, 3 empty test tubes, 3 LB agar plates, and of course, safety equipment. Start by labeling 3 LB agar plates, as shown here. The label should include your initials, the dilution of phage, the amount of the dilution, the type of bacteria, and the date. Make sure a pen is used and that the underside, not the cover, is labeled. Next, label 6 sterile test tubes with the dilutions shown in the procedure. Next, with the use of the micropipetter, aseptically transfer 990 microliters of sterile suspension medium into the 10 to the minus 2 and 10 to the minus 4 tubes. Remember to practice sterile technique, only opening test tubes when needed, and immediately dispose of micropipette tips after one use. For the remaining 4 tubes, the 10 to the minus 5, 6, 7, and 8 dilution tubes, we are going to transfer 900 microliters or 0.9 milliliters of sterile suspension medium into each one. Next, 10 microliters of phage stock is going to be transferred to the 10 to the minus 2 tube. This equals a 100-fold or 10 to the minus 2 dilution. By flushing up and down the pipette tip, we effectively mix the 2 liquids together. Using a vortexer is another technique to mix the 2 liquids together. We will now transfer 10 microliters from the 10 to the minus 2 tube to the 10 to the minus 4 tube. This corresponds to another 100-fold dilution. 100 microliters from the minus 4 tube will now be transferred into the minus 5 tube. This corresponds with a 10-fold dilution. Remember to use the micropipette to flush the liquid up and down in the test tube, as well as vortex after each dilution. Continue these 10-fold dilutions with the 10 to the minus 6, 7, and 8 tubes. On the table are 3 new test tubes labeled minus 6, minus 7, and minus 8. We have here our phage dilutions from the previous step and E. coli. To the test tubes, add 0.3 mils of E. coli and 0.1 mils of the phage dilution. It is very necessary to practice sterile technique and change the pipette tips after each transfer. After the transfer is complete, gently vortex the tubes and set them aside for 15 minutes at room temperature. After 15 minutes have gone by, add 3 mils of melted top agar to the tubes using the power pipetter. Then immediately pour the tube onto the agar plate, swirl around to mix, and allow to sit for 18 to 24 hours. Sterile pipette tips should remain in their packaging until just before they are used. Remember to not open test tubes or agar plate covers until necessary. This will prevent contamination. Set aside these plates for 18 to 24 hours.